Should all infections be forgot and never T-cells primed, will cross-reacting antigens protect the host next time?
This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 422, recorded on December 30th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in New York City on a nice sunny day, but a little bit of a chilly day, Dickson Despommier.
Hello, Vincent.
What do you think the temperature is today, Dickson?
It's about 30 degrees.
30 degrees?
I'd pick that one.
4 degrees Celsius. So you're wrong.
Well,
it's not below freezing.
Always.
You're always wrong?
I'm used to it.
The sun is streaming in now.
Yeah, that's called a greenhouse effect.
And
it's nice and warm.
It says here drizzle starting in 35 minutes.
It's true.
Is that true?
It is.
The storm is coming?
Something's coming. I can see it on the horizon right now. It's a gray presence creeping eastward.
Also joining us from Western Massachusetts, Alan Dove.
Winter is coming.
It is.
Good to be here.
I can see the scythe in your hand, Alan.
Yes. Yeah, it's kind of nice at the moment, but we've had occasional little snow shower clouds blowing through at high speed, gusty winds, and just above freezing. It's 36 Fahrenheit, 2 Celsius.
Are you going to get snow up there?
We got a little bit last night, and we're supposed to get some more, a little bit more tonight, kind of off and on, and then tomorrow it's supposed to be nice.
Right.
Also joining us from Austin, Texas, Rich Condit.
Howdy, everybody.
Howdy.
How you doing?
Howdy.
We got 56 degrees Fahrenheit, 13 Celsius, and cloudy.
Right.
I have to say that in my experience so far in Austin, Texas, no offense, but there's been a lot of this cloudy stuff. And, I find it offensive.
Well, I think you should start to get used to it because it's going to be with you for a long time.
I think that's right.
How about the drought down there? Has that had any relief at all?
Yeah, it had. It was a pretty wet winter, and there's actually been a fair amount of rain since I've been here.
That's
so,
great.
Yeah, they've gotten significant relief.
Because California is actually experiencing the refilling of a lot of those dried up reservoirs.
Yeah, dramatic.
So that's good stuff. Glad to hear it.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
It's cloudy here, and actually the sun is peeking out a little bit, but Rich, I bet we'll have you beat on cloudiness because of where we are in the state of Michigan relative to lakes is that we get a lot of clouds in the winter. So even when it's snowing or when it's not snowing, but there's snow on the ground, it's not the beautiful stuff because the sky's not blue and so on.
Right.
Well, they got no excuse for it to be cloudy here. I don't understand this at all. But
speak to Jerry Jones. Maybe he can help you.
Well, I don't think so.
We'll see.
Kathy, you want to tell us about ASV meeting?
Sure. The ASV meeting will be June 24th to 28th in Madison, Wisconsin at the Monona Terrace, the building designed by Frank Lloyd Wright. And so all of the sessions will be in that one building. People really like that because it makes going from workshop to workshop easy. The things
Even
to
if
think
it's
about.
cloudy.
Yeah. The things to think about now are working on your abstract to submit by the deadline of February 1st, 2017, and your membership to ASV. If you're submitting an abstract, you're abstract has to be sponsored by a full paid-up member, and so a full member that's paid up is what I meant to say. And if you are a student or a postdoc and you want to apply for a travel grant, that deadline is also February 1st, and you need to become a member for that as well. So you can work on your membership now. It's simply your CV and a sponsor's letter. Submit that to the website, and the membership committee looks at it, and then we'll approve it, and then you pay your dues, which is very small amount of money for trainees. And that's basically it. We can talk more about content another time. We've already talked about content some, but basically go to the ASV website to become a member, to see how to do the abstract and all that. asv.org.
Thanks, Kathy. We have a little follow-up from last time. Dickson, take the first one.
Jake says, hello to all and all TWiVm to whom may read this. In episode 421, at about 1 hour, 44 minutes, 34 seconds, Dickson claimed they don't do spaceship noises while in space on Star Trek. And I'm just going to drop this link here. So apparently they do, actually. I'm a mathematician by training who got interested in this whole little danger critter thing back in graduate school when I wrote my dissertation on the mathematics of crop epidemics. And I keep hoping for more plant-related topics on the TWiV sphere. Hint, hint, hint. Thanks for making so many good podcasts, Jake. And okay, so there is a certain soundtrack to Star Trek, which is similar to the...
Did you guys listen to this?
Yeah.
Yeah.
It's very soothing.
And actually it is.
And did you realize that this is part of a series of these videos that are just... They're a picture of the Enterprise, and they're that thrumming engine noise.
Exactly.
Just playing constantly for this one, I think, is an hour. There's another one that's 12 hours.
Wow. For people who have trouble getting to sleep.
And I guess I guess the idea is that you can open this in a tab in your browser and put on your headphones and just tune out the world to the sound of the warp drive pulsating its way through space.
Nice.
Trippy.
Kind of like
Give this a try.
I
think they legalized that in Massachusetts recently.
Now, is this a noise inside the Enterprise or is it outside the Enterprise?
I think it's inside.
So do I, and I still think I'm right that they don't.
I think they play
inside.
it when they may play it when they show the outside of the ship, but I always interpret it to mean that's what it sounded like.
Alan, I'm with you on that. I think that's true.
I still don't recall seeing any explosions that made a sound on Star Trek.
Now, when they blow up the cloaked and then suddenly uncloaked,
right?
It blows up, but you don't hear it.
I just saw the latest one. Rich knows because he saw it too, right?
Right.
Where that bad guy...
Rogue we saw it.
No, that's Star Wars.
That's Star
Trek.
Star Trek.
The recent Star Trek where the bad guy cuts off all the parts of the Enterprise. Right? Cuts off the engines, then he cuts off the main part. Yeah. And that makes noise when he does that.
Yeah, I know.
Okay.
It's not supposed to, of course, unless you're inside.
Did you all see Rogue One?
Yeah, I saw Rogue One.
I haven't seen
I liked it a lot.
I did too.
Not yet. It's on my list for the weekend.
I found it kind of boring.
No, it wasn't boring.
Maybe
great.
I liked
it was like
a lot.
watching a big game of laser tag. I mean, it just didn't really...
I liked it a lot.
I think they should have played it up more as like a heist movie. That would have been good, but
I think it was.
well.
I thought it was okay. So there you go, Dickson.
Yeah.
I knew you were wrong, but I'm finished saying
but
you were
didn't know
wrong.
why I was wrong. That's the whole idea.
Yeah, I know why you're wrong. Because in movies, they make noises. Because if there's no noise, people won't like it.
Right.
So even though there's not supposed to be a noise, or put in your blank there something, they do it just for entertainment purposes.
Right.
All right, the next one is from Anthony, who writes, concerning the discussion of zoonotic disease in TWiV 421, I believe that it's noted in the Natural History of Infectious Disease by Burnett and White that the species for most of its history existed as small, widely dispersed groups. This would mean that, as for pathogens, our ancestors were a market just not worth developing. Disease generally would be zoonotic. The exceptions were vertically transmitted or vertically spread. An example given for vertical spread is kissing of infants by caregivers. Though not Norman Rockwell, fecal contamination is just as apt. This is funny. Taken in with the appropriate antibodies along with mother's milk, these entities could coexist peacefully and perhaps not even count as pathogen. Was it in this way that polio has been with us so long and is the reason why there is no animal analog? To go on to do the high-wire act of taking a speculation as an assumption and then going on to speculate some more, might there be some sort of second amendment in evolution with pathogens as a militia? In both cases, well-regulated is pivotal. Or what's the Second Amendment?
That's right to keep and bear arms.
Oh, that's the one we're about to repeal, right?
Hardly.
Second Amendment.
No, I think that's the one we're about to make mandatory.
In both cases, well-regulated is pivotal. Yeah, I think you're right. I think that humans were in widely dispersed groups, so it wasn't easy for pathogens, at least new pathogens, to spread.
Right.
But the ones they had acquired from their human precursors, or they would have been with them as well, yeah. Alan, can you take the next one?
Sure. John writes, 3 follows up. First, in TWiV 421, an emailer questioned the statement that Earth's rotation has slowed by 6 hours over historic times. The 6-hour figure is not the change in day length, SI units radians per second. It is the integral of rotation speed change over millennia, SI units radians. If Earth had not slowed its rotation since Rome was founded, it would be twisted at about a 90-degree angle to its present position, but the day would still be close to 24 hours long.
That's interesting.
Okay, so this is 6 hours of total time, not our day is 6 hours longer.
Yes.
Total
as
time.
I was listening to you guys discuss this, this is one of these things where I was listening to last week's podcast, okay, and listening to you guys discuss it, and I was shouting at the podcast because I had researched this when we talked about it before, and it's 6 total hours from like 740 BC.
Okay.
Right? Now, the other paper that somebody wrote in about last time was talking about the time change from eons back, some,
right. Yes.
one of those scene eras from way back, and using a totally different set of, totally different bunch of data, okay? Right. They were using physical data of some sort, or atmospheric, or fossils, or something like that to do times. This piece that got the 6-hour figure was looking at historical records of eclipses, of eclipses and stuff.
Right.
All right.
Yeah.
So I think there still may be a discrepancy between the two different measures, but this is still an appropriate correction relative to last time. It's 6 total hours since about over the last 3,000 years.
Right.
And there still may be a discrepancy with respect to the other measurements, but it's a totally different period that's measured and a totally different set of measurements. So who knows?
So I think that Richard triggered a thought in my head as I was listening, and I think that we should characterize what's about to follow politically, at least in the United States, as a new era called the obscene era.
Okay.
I have to agree with Rich that I thought it was 6 hours over time, and so I didn't understand last week's comments. Now it's all becoming clear.
All right. Continuing with John's follow-up. Second, in or near TWiV 419, you discussed inconsistent terminology in a paper. There is a solution used in other fields. Require the list of references to include a review of terminology. Virology papers could state early on, for example, terminology follows Flint et al. 2015. The reader could look there to find out exactly what permissive means. While a top-tier journal might be unable to expand papers by a sentence and a reference, less dense journals could ask reviewers to enforce this practice.
Here on TWiV, we use Flint et al.
Yes.
Right. But the problem with this is that most virologists probably think that they're using the right terminology.
Yes.
I would guess.
Actually, Vincent, is there a compilation of these terms that's a real Flint et al.?
No, there's no compilation. No, could make one.
We need to start one.
It's just Jane's brain, Vincent's brain, now Glenn's brain.
So the funny thing was, at one point when we were writing the last edition, I said I should write down everything that bothers Jane, I didn't do it. It would have been fun because over the course of the 2 years that we were working on it, I would have listed everything.
Well, this would be good. So you'd have the AP style guide, the Chicago manual, and then you'd have the Flint style guide.
Good name. It's catchy, right?
Yes.
Right. Maybe we should do a TWiV 101 on this.
Yeah.
Well, it's scattered throughout the TWiVs, you know. Well, we could do a TWiV.
Should bring
we
them all
could do
together.
a pedantic TWiV.
A pedant.
Right.
Aren't they all?
All right, continuing with John's letter. In TWiV 421, somebody asked, what happens to ads in archived podcasts? Old episodes of the Nature podcast recently got ads that were not present when they were made. I don't endorse this practice, but it is a reminder that history can be rewritten. We've always been at war with East Asia, and Vincent has always been nice to Dickson.
See, Dickson? See?
Always.
Yes. P.S. Here is a vignette on the success of virology. A Chinese friend went to grad school here and did a postdoc in biology at MIT 25 years after Vincent. When she saw my shirt with a vaccine bottle on it, she read it and asked, "What is poliomyelitis?" My explanation may have been the first time I said polio out loud since high school, and she hadn't heard it in 16 years in America. I thought to myself, previous generations of scientists did such a good job, their reward is being forgotten.
No, the reward is being forgotten.
Yes.
Rather than the reward is being forgotten.
It's not like it's not in the news, so I'm really surprised. Right, because apparently
it is in the news.
From time to time,
Sure, it's in the news.
A little bit here and there. Yeah, so sure. I'm not really sure why a biology
and
post.
If you travel to other countries, you can find it all over the place,
course.
especially India and places like that. So.
Absolutely. By the way, this business with old podcasts getting ads, one way that could happen, what's the Stitcher? Stitcher inserts ads into our podcast that we don't put in.
Huh.
And also Google Play does that. Just randomly.
Oh, really?
So they can take if you put your podcast on Google Play, they'll take all your content and put ads in it.
Goodness.
So they weren't there to begin with, and I don't like that because I don't want them to interrupt. All of a sudden we're being pedantic and they interrupt us. How could they do that? Right?
That is really not cool.
It may not be Stitcher, but I know it's Google Play. So that could be what's going on there. If Nature actually went back, and it's a lot of work to do that, so I doubt that they did that. But Google, of course, can do everything automatically.
It's true.
Okay, Rich, I think you should take next one.
Yeah, okay, so here we go with the Earth again. John writes, Dear Vincent and TWiVestigators. That's pretty good.
Not bad.
There seems to be some confusion about the Earth's rotation is slowing, picked from TWiV 420. The authors mentioned that their analysis found an increase in the length of the mean solar day at an average rate of 1.8 milliseconds per century, which is less than the predicted figure of 2.3 milliseconds per century, according to some previous data. To simplify slightly, right, the difference between the length of a day now and the length 1,000 years ago is on the order of 20 milliseconds. However, the day-by-day cumulative effect of each lengthened day is about an hour for 1,000 CE and 7 hours for 500 BCE, according to their equation 1.5. A day today and a day 2,700 years ago have slipped about 7 hours out of sync if you compared the position of the rotation of the Earth versus the position of the Sun, I think. Because they can calculate the start and end times of ancient eclipses with a high degree of confidence, and they have ancient local records that indicate when they occurred in local time, they apparently can use the difference between historic and calculated figures to extract some information about what the actual change in the spin rate is. Well said. The historically measured rate changes differ from the rate changes given by the theoretical tidal model. None of this really contradicts other studies, and he quotes one, using daily and annual growth rings on ancient fossilized coral to calculate ancient day lengths and the larger number of shorter days in ancient solar years. It just adds some more direct data into the mix. Radiolab had an excellent 13-minute piece explaining about the coral Daylength discovery with paleontologist Neil Shubin, Fields Museum's chief curiosity correspondent Emily Grassley, and Fields Museum's collections manager Paul Meyer. I submit it as a listener pick, so let's keep that.
Nice.
And quotes, we discovered that our world is full of ancient coral calendars. Each one of these sea skeletons reveals that once upon a very long time ago, years were shorter by over 40 days. And astrophysicist Chris Impey helps us comprehend how the change is all to be blamed on a celestial slow dance with the moon. I haven't had a biology background, but I love the Twix family. I'm going to attempt Vincent's virology course in the new year just to get a better handle on the language of the podcast. Mm-hmm. And that's from John, who's in Limerick, Ireland, which is relevant because he says, as a PS, the forecast today is for unseasonably pleasant, mostly dry with sunny spells in places through, though cloud will increase and produce some patchy drizzle near coasts later on this afternoon. Highs of 7 to 9 degrees C with light south to southeast winds, 10 to 20 kilometers fresher on the coast. P.S. Be nice to Dickson. And I thought he had said somewhere that ironically,
where'd it go?
Although he's from Limerick, he didn't submit a limerick.
Yeah, where did it go? I have to find. It was right there.
Has anybody ever read the book The Limerick?
John, let me look. No. Let me look up his email.
You have children and grandchildren. Don't pick it up. I made them one of my major parental mistakes by buying that book because we all liked to make little rhymes. So I picked up the book The Limerick, thinking it was just another collection of short. Well, every one of them. I would start to read it. I couldn't get past the second line. And all of them rhymed with every part of the body you could possibly think of. So it's called The Limerick, in case anybody out there wants a good laugh, but an adult laugh, not a children's laugh.
So Emily Grassley has that YouTube channel that was picked on TWiV by a couple of people. What's the name of it? Just really good videos.
It was also picked on TWiV. Or no, TWiVo.
TWiV. And in fact, her colleague at the field
was on the TWiVo.
Picked it, and I'm going to look at it right here. Yes, Corey Moreau, who we had on TWiVo 13. She's a entomologist. It is Brain Scoop. Yeah, there you go.
Brain Scoop.
I think Alan picked it also, maybe.
I may have.
Brain Scoop. I love it. She's the chief curiosity correspondent at the museum. Now, Neil Shubin is going to be on TWiVo next year sometime.
Nice.
Another cool one. And the last email, Kathy, please.
Michael writes, enjoying your podcast here in Rochester, New York. Temp outside is 0 degrees Celsius, light flurries. On a recent podcast, someone raised the question of inapparent infection in mumps, given the recent outbreaks at SUNY New Paltz and SUNY Geneseo. We received a recent alert from our local health department regarding diagnosis of mumps, and this tidbit came up thanks to Up to Date. Inapparent infection. In contrast with these classic manifestations, asymptomatic infection occurs in 15 to 20% of cases, and only nonspecific or predominantly respiratory symptoms are seen in up to 50% of cases in whom the diagnosis of mumps is not usually made. Inapparent or subclinical infections are more frequent in adults, while parotitis is most common in children between the ages of 2 and 9 years. Happy New Year to all of you, and I really enjoy your podcasts. Michael, and he's a physician in Rochester.
Right.
All right. Now, this is the last episode of 2016.
Indeed.
So in following a tradition that we have followed for many years, I think since the beginning, except that first year.
I think so.
We do a kind of year in review. And let me give you some numbers before we tell you about our 10 favorite topics. Now, we released in 2016 53 episodes from 370 to 421. This one we're doing today is going to be released next year. We did 7 shows on the road with guests, And we had done 5 the year before, so we did 2 more. And last year we had 25 in-studio episodes with guests. And the previous year we'd only done 4.
Wow.
So we really did more. And that was a goal at the end of 2015. I wanted to have more guests, so we achieved our goal, which is cool. I don't know how we can get better than 25. That's a lot.
That is a lot.
It's almost half. It's almost half of the episodes. And then we did also 7 episodes with video last year. That's down from 13 the previous year. And, it'll probably stay the same. If you're interested in our total downloads, for 2016, 832,892 downloads of all the episodes, those 53 episodes, ranging from about 60,000 to 70,000 a month. The highest month was February, 73,673. Of course, those numbers will keep increasing because as people go back and listen, they're added to the tallies. Yep.
February is when we covered Zika on at least 3 of the episodes.
Yep, the Zika bump. There you go. The biggest episode in terms of downloads for 2016 was TWiV 375, Zika and You Will Find. That got 18,222 downloads. And by the way, all-time TWiV, we're well over 5 million total downloads from the very beginning. That's pretty good.
That's great.
Yeah.
A lot of listens.
Indeed.
I also wanted to tell you a little bit about who is listening or where that is. Of course, the country with the most downloads over time, 2.8 million, is the U.S., followed by the United Kingdom with 383,000. So there's a big disparity there. Mostly in the U.S. people are listening. The third country is Canada, then Australia, Germany, China, Nigeria, Sweden, the Netherlands, and Japan.
I bet Nigeria per capita is the highest.
Probably.
Yeah. That's amazing.
Yeah.
Actually, I don't know the population of Nigeria.
It's 75 million. It's about
Nigeria is
75
pretty
million.
populous.
75 million?
It's up around that.
But still, that's... I'm impressed with the geographical diversity. That's cool.
That's pretty good.
Yeah.
I mean, these are only the top 10.
Right.
Japan there is at 37,000, so it's not a lot, but it's still great. I'm impressed, and we can still gain more listeners overseas.
So most of these countries have a high priority for learning English or knowing English, and it's obviously understandable that Japan is low down because they don't emphasize that in their culture. But still, you've got 36,817 listeners. That's great.
Now, in terms of regions, the top region is California in the U.S., 420,000. So, that's within the U.S., which has the most downloads. It's California. Then Virginia is number 2, which is interesting. Then New York.
Yeah.
Then Missouri and Texas. Now,
Dang.
Virginia's interesting because I wonder if it's because so many people from NIH live there.
Right.
But I don't know. 225,000 is way more than NIH employees, so it's got to be something else. I don't know what.
Yeah, and it's not. I mean, California at the top makes sense because it's a big population.
Yeah.
There, but then Virginia is an odd one for second place. That's interesting.
Of course, Texas is another one you expect. Now that Rich Condit has moved there, it will even get bigger.
That's right.
But below Texas, Washington, and then we have Hartford in the UK. And then we have unknown in the United States, which is actually a lot, 116,000 people who they can't figure out
where they are.
They
keep moving.
Massachusetts because of Alan Dove.
Oh, well.
And then Maryland.
Wow.
So Maryland being 10 and Virginia being 2, maybe that means there's a lot of policymakers listening.
Could be.
Wishful thinking.
Yes, it's nice to dream.
Anyway, let us know what you want to hear as always. We know we want to hear more plant-related virology, but what else? And so we have people to thank for this year. Jennifer does our timestamps. We have to thank her. Also want to thank American Society for Microbiology and the American Society for Virology for supporting live episodes, which we did last year, which we will do next year as well. And, of course, we can have more of the team join us in these live episodes because of your support. And, in fact, at ASV, we hope to all be there, or at least most of us. Yeah. PLOS, Public Library of Science, supported our episode 400. We thank them for that. We also want to thank the universities who hosted TWiV University of Pittsburgh, Penn State, Boston University, and University of North Carolina. There'll be more of those next year.
Mm-hmm.
Of course, we want to thank all the listeners, those people who have led to these great download numbers, which I think, all the podcasts we do, these are the highest, followed closely by TWiM. And of course, I want to thank all of the people who have been on TWiV, as well as Dickson and Rich and Kathy and Alan. Thank you so much for doing this. I appreciate the time spent.
It's an effortless thing for us to do, as long as we're in town or can do it.
For you, it's clearly effortless.
Put this right at the top. Thanks.
I don't know about
I make it sound so easy, right? That's the whole deal, right?
The fun payoff is well worth it.
My New Year's resolution is to be nice to Dickson.
I tell you, it's interesting because when I go home after this broadcast, or podcast, I should say, and my wife confronts me at the door in a pleasant way. Of course, she says, "Okay, what was TWiV about today?"
Really?
Yeah, she actually asks about it, and
well, you can't answer, of course. Right?
Well, sometimes I can. Sometimes I say, "Well, it was kind of an interesting paper," and then But one of them related to an episode that we could relate to my grandson because he had a rash that was a virus origin, and we had to look it up and go through all kinds of things. So it raises interest.
Dickson, do you tell her I'm nasty to you?
I don't say a word about that because you're not. You're not nasty to me.
Well,
Vincent.
people seem to think
no, but you're not now. I can honestly say that it's not done out of any other than a spirit of having fun. You want to taunt and all that stuff, but it's all done in the tongue-in-cheek type of fashion. I don't take it seriously.
You can tell your wife, Dickson, that sometimes when it's over, I don't know what TWiV was about. And I want to echo Alan's comment to thank you, Vincent.
Yeah, absolutely.
You're the guy who makes all this happen.
Could not happen
without you. This party.
It's a high point in my existence.
And he does all the work. Trust me, he does all the work.
Oh, cut it out.
Yes.
He does cut it out?
Well, it wouldn't be a show without everybody participating.
Yeah, but you do all of the fine details that make it sound professional.
Yes, and I will thank you too. It's just really fun, and I've had way more than my 15 minutes of fame.
Yeah,
so
we can be famous for a long time.
Well, you deserved it.
And so we're going to continue, of course, in 2017.
Absolutely.
And because it's a lot of fun, and we know you like it. So, and we also appreciate your support. Those of you who have supported us on Patreon or in other methods, we appreciate it a lot. And we're going to have our last advertisement for a while today. Most of these advertising companies don't advertise in January. I think they figure people have spent all their money.
That's right. They're in recovery.
Maybe we'll have some new ones after that. But we've had a long run with CuriosityStream. 6 months is a long time, and we appreciate that. But they said we are a really, really good performing network for them. They really were happy about it. So thank all of you. But I do think that it's a good match for TWiV, right? The science documentaries and so forth. So we have 10 topics, virology stories from last year. No particular order. We'll go through. Of course, the first one that comes to your mind is Zika virus, of course, which we have featured on many episodes.
Many episodes. I was going through the back episodes to prepare for this, and I thought, I got to mention what episodes we covered Zika on, and I gave up. I see somebody managed to list all of them, right? But yeah.
I listed almost all of them. There were one or two where maybe it got 30 seconds of discussion and I left that out.
Well, and we did, the Zika sit rep has been a fixture of the show pretty much all year, so.
So the first time we covered it was in 2015, December 20th. And then again in February. Of course, in the beginning of 2016 is when really it had already been infecting in Brazil, but then the connection with microcephaly was a big concern. And of course, initially it wasn't clear if it was an association or causation, but all the data that came out over the past year made it quite clear that infection during a certain time in pregnancy leads to birth defects of various sorts, not just microcephaly, but other things as well. We have a lot to learn.
So
right,
Vincent, do you remember when it first? Does anybody remember when it first came up that the connection with microcephaly as a possibility?
Well, of course, in Brazil there was an increase in the number of cases in that northwestern state.
Yeah, I'm just wondering what time. So
Early.
We were talking about it before that actually came up, right?
I think it was February or March, wasn't it?
No, I think actually in December of 2015, we actually knew about the microcephaly.
Okay.
Yeah, I believe that's correct. Because remember, it seems to have, people started noticing an increase in rash in the beginning of 2015 in Brazil. So for the microcephaly, you need 9 months.
Yes,
right?
on this December 20th thing, there's a link to microcephaly in Brazil. Okay.
Right, okay.
Yeah. But, we were skeptical at the beginning because it was an association, but then lots of data from human cases, finding virus in fetuses, damage in fetuses, animal models which provided more evidence for infection. And we had lots of guests on the show talking about it, right? We had Carolyn Coyne, Helen Lazier, Esper Callis, a physician from Brazil, joined us, Sharon Cern, Scott Michael. Is that everyone? Michael Diamond, of course.
Did you mention Jeremy?
Jeremy Luban.
Jeremy.
We had Michael Diamond, right?
The ASM ASV meeting June 1st.
Right. So that was Michael Diamond, Charlie Rice, Laura Kramer, and Michaela Gack. Wow, I remembered all their names without looking at it. I'm not gone yet, Dickson.
No, you're not.
So I have here at least 24 episodes in 2016 with some Zika.
Nice.
And if you count the Zika sit rep, maybe more than that.
Yeah.
So what is the status with Zika? So I was looking to see, are there still cases? Because it's really not much in the news anymore in the way it used to be, of course. It's no longer news. So there's a cool page at the European Center for Disease Prevention and Control where they have a map where countries are colored whether there is not much going, no more than 10 locally transmitted cases, more than 10 cases currently, or no transmission. So apparently, parts of Southeast Asia and Central America, Caribbean, and South America, there's still ongoing transmission. Now, it goes down there in the south. It's summer, right? South of the equator, so that's not surprising. But Dickson, you were saying something about it being dry now, and that's limiting the transmission.
It is dry, but I also mentioned the fact that it was hard to find anything in the U.S. news that talked about it, because I think that there's been some pressure from these tourist destinations to play down the fact that they've got Zika there, but they've got a lot of other things there too, and they don't talk about those either. They don't talk about chikungunya or dengue, or they've got malaria or filariasis, and still people go down to Haiti and they go to the Dominican Republic on vacation.
Yeah, but they don't cause birth defects.
No, that's right.
So, and they're not sexually transmitted, so this is the big problem.
But the tourist industry has really fallen off the edge. I mean, it's really taken a nosedive.
Well, I think
this has really especially hammered Puerto Rico at a time when they were not in good financial shape, and then the Zika fears. They've done huge ad campaigns talking about how it's safe to come to Puerto Rico, which of course that just makes people even more leary of it when you tell them that it's safe.
That's true, and I think the Virgin Islands had some big hits.
Yeah. And it's just it's a serious blow to these places.
Yeah.
And I would go to those places.
Because we're not going to give birth to a microcephalous kid.
Right. I would say if you're 2 months pregnant, you're thinking about going on vacation, maybe don't do the Caribbean this year.
Right.
But if you're not, if you're part of the majority of the population that is not currently pregnant, then these destinations are fine.
A few other things about Zika. I found just today an article in PLOS, Neglected Tropical Diseases, a paper entitled Prevalence of Antibodies to Zika Virus in Mothers from Hawaii who Delivered Babies with and Without Microcephaly Between 2009 and 2012. These are stored serum samples, which they went back and they tested. And they found both IgM and IgG antibodies. Now, remember, I think it's hard to distinguish Zika and dengue, right?
Right.
Is there dengue on Hawaii?
Good question. The answer, I think, is no.
Let's look it up.
But
dengue outbreak. There was an outbreak in 2015, according to the Hawaiian.
Yes.
So there is there. So that could be complicated. Anyway, they say that the presence of antibody positivity to Zika showed a trend towards significance with microcephaly. Antibody positive mothers were more likely to deliver babies with microcephaly than mothers who were negative.
The dengue story in Hawaii. I'm now reading this Department of Health page. It's kind of interesting. Apparently, they do not have endemic transmission of it.
Right.
No, these are all
so it's
travel.
Just occasional imports that then set up a local transmission cycle. Then when that burns out locally, this 2015 one was on Hawaii, the Big Island.
Got it.
Then there apparently one was in 2011 on Oahu. So periodically one gets imported, and then there's a local transmission cycle for a few infections, and then it dies out, and then they don't have dengue anymore.
Yeah.
It's not locally sustained.
Right.
Yeah, and that, yeah.
So they have Aedes aegypti there, though, clearly.
Yes,
they do.
Anyway, this suggests that there might have been, there was Zika on Hawaii, I think, unless these are all wrong and dengue cross-reactivity. But in 2009, and of course the Yap Island outbreak, 2007, followed by outbreaks in the Pacific, so it could have gotten to Hawaii that way.
That's true.
Right, so that's interesting. The other article I found that's of interest is an article in the Times today. They occasionally still publish articles on Zika. It's called a Zika vaccine, but for whom? Whom, Dickson?
I heard whom. I agree with the use
So
of
we
whom.
may have a vaccine by 2018, but who is it going to be for? If history is any guide, impoverished communities are likely to be last in line.
It's true
And
for all
so,
vaccines.
I think it will certainly be a travel vaccine for the U.S., but
Absolutely.
yeah, for other countries it should not be. But who's going to pay for it? So that's an interesting issue.
Anytime you say who's going to pay for it, I would say yes, because WHO should pay for it, but they don't.
Well, they can get donors to pay for polio vaccine, right?
They can, or they can get companies to donate. Compassion.
Well, for example, many doses of the Ebola vaccine have been donated and stockpiled.
Sure, exactly. So that's an ongoing behavior.
So the rest of the article talks about some of the things that have been learned, and it's actually quite good. I thought
it was good too.
And one of the issues, one of the authors is actually a medical student or a student at Harvard Medical School. I don't know what kind of student, it doesn't say. So that's kind of nice that they have someone who might know some science. But basically, they go through a number of the things, and they say, we thought that it may have changed from Africa to the Pacific to Brazil, but it looks like all those strains can infect the brain and cause problems. So maybe the virus is not substantially biologically different. So it's actually good in that sense. But I do want to make a little pedantic rant, not too much, but here's a quote, "It may only recently have evolved the ability to cause microcephaly and brain damage in babies." So that's what they go on to say is likely not true. But viruses do not evolve abilities to do anything. Okay, evolution is a random thing, and I don't.
Yes.
If you talk to the authors about this, they probably would understand. But when it's easier to write it this way, right? But it's not right to say they have evolved the ability. It implies some kind of intent, which is certainly not the case.
Or directional.
And it implies that this is an ability.
Right.
An ability. There's an implication that there's something useful about it, whereas
that's right.
I can't see this as anything but an evolutionary dead end for the virus. I mean, it doesn't. Getting to the brain does not help it get into the next mosquito to reach the next host.
Sure. Absolutely. Right. So there are two problems. One is that evolution is random. It's just mutations emerge and the right selection conditions select for them or not. And then what you say, getting into the CNS is a dead end. So
right.
Yeah. So I was unhappy about that. All right. So Zika is a big one. Any other comments about Zika? All right. By the way, if you want a Zika TWiV mug, I made one. It's on cafepress.com. You can find it. Someone said I should make special mugs with different viruses on them. So I got Zika. I'll make some others. So the Zika was easy because I had made the image of.
I was going to say, in your free time, you could probably come up with all the viruses.
Yeah, I borrow time from other people. The next one that I stuck in was Ebola virus, which wasn't in the news all that much because the outbreak was over.
No, it was pretty much
year.
It was pretty much done by the time we started. Yeah.
Yeah. So, but we did do a couple of things. One of them, not too long ago, was an episode with Jeremy Luban and Ted Deal and someone from the Sabetti lab. Let's see. I should get this up here, and we could give him credit. That was Aaron Lynn.
Mm-hmm.
And, this was some evidence suggesting that a particular change in the glycoprotein of the virus increased infectivity in human cells.
However, as you so aptly pointed out in the last comment that you made, the virus itself didn't adapt to humans, but rather was randomly selected for life.
Well,
because
that's the question, is whether it had anything to do with people, and that's hard to know, and we had a discussion on the episode about that, which I think is very interesting. And, if we ever do see another outbreak of Ebola, I hope we never do, but if we do, it will be interesting to see if other changes, this one or other changes arise and are selected for that affect infectivity. Because all you can do is look at human cells, and animal models don't really tell you what you want to know.
Right, and this particular epidemic was really, really exceptional in scale.
No question.
And gave a lot more evolutionary time that the virus spent in humans and transmitting human to human.
Yep.
I certainly hope that we don't have another epidemic of that scale that would permit that type of adaptation.
Yeah, not. I think we probably won't, because we'll be more prepared, right?
I like to think that maybe the world is a little better prepared than that, but there are a lot of things that I like to think that don't work out. So
all of the hyperbole associated with this thing going global from the middle of that outbreak is just, it elicits smiles on my face, because that happens every time there is a human crisis going on someplace, and they tend to globalize the events and make them bigger than they really are. This was big enough without having to do that.
Yeah.
By the way,
one of the things I liked about
Go ahead.
One of the things I liked about that episode was the sort of serendipitous parallel work from the two labs that were represented on the paper, and how they were looking at different features, and then realized that they were working on this parallel but complementary path.
Yes.
And brought it all together. It was really cool.
Yeah.
By the way, the episode image, there's an image with every episode. Many of you do not look at the website, which I fully understand. But I try and find a cool image. And this one I got from Ted and Jeremy, which was not published. I can't just take images from published papers unless they're open access. So they sent me a cool one, which shows this change in the glycoprotein. There's a little map showing how it spread throughout the three countries from April to July to May of the next year. And then below are some experimental results in cell cultures from humans and mice and dogs and cats, showing only in humans you get higher infectivity. There's cute little cats and dogs and mice. Anyway, I like picking these images. So another Ebola story we mentioned last time. The final results of the ring vaccination trial have come out. This is something we covered in 2015. They had a preliminary report. The final article is in The Lancet. Efficacy and effectiveness of a recombinant vesicular stomatitis virus vectored vaccine in preventing Ebola virus disease. Final results from the Guinea ring vaccination open-label cluster randomized trial. Ebola ça suffit. It's enough. So this is a cool approach to, first of all, it's in the middle of an outbreak, which is tough to do, to test a vaccine, right? And you don't want to give people placebos, right?
And it's not just in the middle of an outbreak. It's in the middle of an outbreak that's happening because there's no functional healthcare system.
That's right. Mm-hmm. So this ring vaccination design, which was the advice to do this, one of the advisors was D.A. Henderson, who passed away last year. They would identify a case of disease. Then they would make a list of rings of people, a cluster of all their contacts and contacts of contacts. And then they would randomly assign these clusters to either immediate vaccination or delayed vaccination 21 days later, and they would look at the incidence of Ebola in them. And essentially, they looked at a lot of people, 4,500. They rolled 4,500 contacts who got immediate vaccination and 4,500 who got 21-day delayed. The numbers are actually lower because not everybody consented. And in the immediate vaccination group, 0 cases of Ebola. And
after 10 days.
After 10 days.
After 10 days, right.
The assumption was that within the first 10 days of them being vaccinated, they could have already been infected. And so those infection rates were the same between the delayed and the immediate vaccination groups.
Right.
And then the delayed group, 23 cases.
Right.
As I recall, they also had a sort of an internal, a lucky internal no vaccination control because of people who didn't qualify or who didn't consent to the trial. They could follow them as well, which was interesting.
So they said the vaccine efficacy was 100% based on that number.
Right.
Well, the WHO said it could be up to 100% effective. And in fact, somebody that was interviewed, I just wrote this up because I'm going to do it as a short news thing at the first class of virology next week. Where's that caveat? Rats. There's some statistics. Oh, yeah. There's a 90% likelihood that the vaccine will protect in more than 80% of the cases. So that's the extrapolation from the data to a larger set.
Right.
Based on the sample size,
and Tony Fauci was being interviewed on NPR and had to explain that difference.
Right.
Did he?
Yep. He did, but I'm sure half of the audience didn't quite catch the math involved in it.
So another thing that interests me about this that I want to point out is that this now is another example of what is becoming is rare but becoming more common of using a recombinant vaccine. So this was VSV with its glycoprotein substituted with the Ebola glycoprotein. The only other examples I can think of are what's it called, Dengue, which is the yellow fever backbone with the dengue structural proteins put in it, and those are the only two. That's the only licensed recombinant vaccine for humans. This Ebola one is another example, and then they've used some recombinant poxvirus vaccines in trials for HIV that have met with limited success. But I think we're going to see more and more of this.
For sure.
In fact, they mentioned one that's under development, or I think we've already heard about this, the Ebola glycoprotein in a chimpanzee adenovirus. That would be the first dose, and the second dose will be the glycoprotein in the pox MVA weakened pox virus backbone.
So this shows that you can do a trial in the middle of an outbreak and where there are lots of problems. And this is the vaccine that's been stockpiled, as I mentioned earlier. So if there is another outbreak, they will be able to deploy it. They don't have enough to immunize entire countries, but I think they have a few 100,000 doses. But you can certainly immunize healthcare givers and contacts of contacts and so forth.
Right. You do the same ring vaccination approach.
Yeah.
One ring to vaccinate them all.
One ring to vaccinate them all. Did we use that as a title?
I don't think so.
Sounds familiar.
It does sound very familiar.
The Ebola vaccine only rings once.
And we also had a Ebola theme or centric TWiV in Boston. Was that in Boston? Yes, because Alan was there. TWiV 394 was at ASM Micro with Erica Ullman Sapphire.
TWiV 349, we did use one ring to vaccinate them all. Okay.
Was that this year or the previous year?
That was
August 9th,
2015.
2015. One ring to... Yeah, that's a good one.
That's
And that's
that,
the one
and
with
that
your
is in
license
fact
plate
the
as
same.
the show image.
Yes, your license plate is there.
No, it's not his license plate. It's Isabella's.
Right. Oh, right.
Isabella's.
Yeah, it's from Ohio.
ESV, baby. Yeah.
Somewhere in Ohio, right?
Toledo.
Toledo, right?
With the frame that says "Crazy Scientist." I love that.
Mm-hmm.
Yeah, that's a good frame. All right. I also thought that a cool episode was 412, the finding of eukaryotic genes in a bacteriophage that infects Wolbachia.
That was pretty cool.
Of course, is a endosymbiotic bacterium that lives in many, many insects of the world. And in other stories that we covered, can be used to put them in mosquitoes, and hopefully they'll be resistant to different viruses. But this is very cool. They finally sequenced the entire genome of this Wolbachia phage and found eukaryotic genes from various sources, including spider venom genes, which I thought was really cool.
Yes.
And other things as well. And the neat thing is the open reading frames are there, which suggests that the proteins are functional and confer some advantage. And I think that's going to be some interesting future work from that laboratory. Seeing where these genes are coming from and what they're doing, So I like that a lot. I thought that was cool. And if there is an earlier TWiV with Seth Bordenstein, TWiV 332, where he talked about his work on this phage, which is called WO, W-O. And of course that was before they had the sequence, but you might want to check that out. Back to Ebola. I forgot to mention Boston Quammons.
Oh, yes.
We talked about Ebola there because, of course,
we did.
David Quammen has written a book about it, and he talks a lot about slogging through the jungle and looking for
that was a fun interview.
Ape poop and narrowly being missed by charging elephants. It was really interesting, so you should check that one out. He talked a lot about Ebola there as well. We had a lot of guests. So number 4, we had it would be all of our guests.
Yeah.
But right at the top is Harold 400 Varmus.
That's it.
Which we did here. Everyone was here.
Yep.
Yes.
That was a gas.
That was an amazing episode.
really
I
good.
My
thought.
all-time favorite.
He was a great guest.
He was.
It would have been even longer if he didn't have to leave.
Yes, exactly.
Yeah.
We could have gone
on forever.
He would have, because originally he said, "Yeah, I got all day, no problem." And then it turned out he had to go to the Democratic National Convention in Philly.
That's right.
And so he kind of rushed out at the end. But that's a great show. He's so smart and so eloquent. So having everyone there was cool. So, we'll do it again for 500.
Absolutely.
Somewhere in the world. You can bet we'll all be there with your support, of course. So your support made it possible. Also, Plas helped us a lot with that episode as well. Who else did we have over the years? Stephen Russell. That was at ASV. Does anyone?
I really like that one.
That was called.
I really enjoyed him.
That was called the Cancer Thief. Does anyone know why I called it that episode? That title?
I have no idea.
No, huh?
Maybe that was that could be that was some quote from Russell himself. I don't know.
No,
Right.
No, it wasn't.
Right. I mean, it's about oncotherapy and.
but why thief though? Because
I don't know.
Here it is, the movie The Bicycle Thief. Has anyone seen that?
No.
Does anyone even have heard of it?
I've heard of it.
All right. I'm going to tell you exactly. It's really a lovely movie, but extremely depressing. It's directed by Vittorio De Sica, 1948. All right, it's a black and white film. Takes place in post-World War II Rome, and I love it. It's great. So that's why I called it The Bicycle Thief. The Cancer Thief instead of Bicycle Thief. David Quammen, of course. Carl Zimmer was a good one.
He was great.
Stan Lemon, Erica Ullman Seyfire. I mentioned Peter Palese, Sandy Weller, Michael Diamond, and many others. We had lots of guests.
Couple sets of guests multiple times. So Jeremy Luban was on 4 times. Helen Lazier and Mike Diamond and Ralph Barrick were all on 2 times each.
So. Right. So some of them were here in studio, some of them were via Skype, and some of them were at meetings and so forth. So we'll do more of that because it's always interesting to get other people on and actually hear how the experiments actually went in what sequence, not the sequence shown in the paper, right?
Right. And that list wasn't comprehensive because there were a couple of herpes virologists we talked to and so forth. But,
for sure.
Lots.
Number 5 is vaccines. Talked a lot about vaccines.
We did.
1, 2, 3, 4, 5, 6, 7, 8 episodes I have here. But probably even more.
About
could be more.
Vaccines and Ebola vaccines, but also just vaccines in general. There was an episode discussing the movie Vaxxed.
That's right, Nina Martin, right?
Nina Martin, yeah, talking about she took one for the team and actually sat through that and talked about the culture of that audience.
The last two episodes, we've had this arc of new infectious but replication competent vaccines.
Cool new technologies. Yeah,
new technologies, which we like.
I was really taken with the nonsense mutant suppressing cell lines and the ability to make mutants that have stop codons in them. I just thought cool. That was terrific. Yes. I think that's got a lot of potential.
Yes,
not just for vaccines, but for doing virus genetics in general.
A couple of people have written in and said, these are not new. Lots of vaccines replicate in one cell but not another. Yeah, but generally they were selected, but these are engineered to be that way. I think that's why these are cool.
Well, and the nonsense mutant, it seems to me at this admittedly very early stage, but it seems like, as Rich just said, that could be a really good general purpose platform for a whole bunch of things.
Yep. So we'll see. They've been tested in mice and non-human primates in these last two papers. Right. So we'll see now if they move into people, clinical trials. We'll see what happens.
And if people use these same approaches for other types of experiments.
Yep. Number 6, mutualism. We did a number of episodes.
I don't know that all of these are actually. I don't know that all of these are actually mutualism, because I'm not very flinty with these sorts of terms.
I'm not very flinty,
but
it's a new term.
But they have. They all talk about viruses involved in some sort of multi-organismal interplay where there are complex relationships.
Yep.
One of them was the what's the creature that Cyclops? Is that it?
Cyclops. Yep.
The zooplankton. Not plankton. What is he?
It's a crustacean.
What? Crustacean. So, Cyclops that eats blue-green algae and poops out the viruses.
Yep.
Actually, eats paramecia.
Paramecia, right?
And that contain the blue-green algae.
Yep.
That contain the virus.
Yeah.
I'm screwing this up, but at any rate, that releases the viruses.
So, Rich,
or otherwise,
if you want a simple definition of mutualism, it's just that each of the organisms that are in contact with each other mutually benefit each other's biology. That's all. That's all it means.
All right.
Yeah, I guess
so at any rate,
the predators are not getting. Well, they're getting food, I suppose, and then the virus is released.
That's kind of a passive mutualistic behavior.
Right.
But I think when you think of a true mutualistic behavior, you think of lichen, for instance, which is a fungus and an algae that live together, and neither one can live apart. They need each other. So if you have a sign on your things that says, microbes, we need them, and it should also say, microbes, they need us.
Absolutely.
So when you put those two things together, that's a mutualistic behavior.
I guess you could call. All right. So then there was the Matthias Fischer, Thomas Hackle paper on integration of the virophage Ma virus into the host, Cafeteria roembergensis. So Matthias actually called that a mutualism because remember the release of those viruses protects some of the cells. So the virus has a host to live in, and the cells are protected. It's a kind of mutualism. I think that would fit, and that's part of this whole theme here. I also like, TWiV 402, cucumber mosaic virus infected tomatoes attract bumblebees. That's pretty cool.
Just crazy.
They get pollinated, right?
Yep.
And the bee gets that. Dickson would call that passive.
It is.
But they call it a mutual benefit here. We appreciate that enhanced pollinator service for infected individuals in wild plant populations might provide mutual benefits to the virus and its susceptible host. You get the point. You get the idea, though.
Yeah.
Great show title, too, The Plight of the Bumblebee.
Yeah.
The
Yeah,
plight
exactly.
of the bumblebee. Absolutely. Alan, you cannot leave TWiV because otherwise we wouldn't have
I'm
any.
not sure. Was that one mine?
That was mine.
Was
That was
yours?
Dickson's.
Wow, you
Yeah.
can- you
Oh,
can-
Dickson.
Be nice to Dickson.
Great.
Now, what's this YouTube video referring to?
Oh, that's a Dar Williams song called "Flinty Kind of Woman." So for your listening pleasure later.
And Flint is certainly a Leadville type thing right now, too. So watch it.
Right. Yeah.
Kathy, that's not too far from you.
Leadville?
No.
Flint.
Flint.
Flint. Oh, Michigan. Sorry.
Yeah.
That's what it, yeah.
We did a.
Jane must be aware of her role here on TWiV. Is that right?
I don't
Does
think she.
listen?
No, I don't think she listens. No, a lot of people don't listen. Although I'm always surprised when we get a letter from someone. Yeah, I say I can't imagine that they would listen, and they do listen. So,
Yep.
but I don't think Jane does. No, I don't know why, but it's fine. Let's see. We did a bunch of episodes on mosquitoes. I think a cool one
Lots.
was 390, building a better mosquito trap.
Yeah.
Yeah.
That was really fun.
The folks from Microsoft building these traps that are flown in by drones to
Yep.
capture mosquitoes and find out what's in them. Although, of course,
Well, the cool thing was that they had these, what were they, infrared sensors or something like that on these little doors on the trap sensed the insect's wing beat and would either capture it or reject it based on a decision as to whether or not it was a mosquito.
yep,
Yeah, it's a smart
Oh,
trap.
awesome.
it's very cool.
Yes.
Yeah.
Right. And the other thing about that was that you could then analyze them, find out what viruses they're carrying and get a lot of information about what's spreading and what's in the area beyond just what species of mosquitoes are there.
Of course, as some people then subsequently pointed out, once the mosquito has a blood meal, it doesn't want to feed for a while.
Right.
So, you're getting here hungry mosquitoes, I suppose, right?
Right.
Exactly.
And so, but maybe they still have viruses in them. I'm looking forward to them publishing this at some point. We had two guests from Microsoft on that. That was cool. But we also talked about mosquitoes in other ways. In other episodes, we had Laura Kramer on TWiV 392, and she, of course, works on mosquitoes that vector West Nile and now Zika, and she was really interesting. And then we talked about using Wolbachia to inhibit Zika replication. You put Wolbachia in your mosquito, you release them, and they don't replicate the virus very well. TWiV 388, what could go wrong?
Yes.
What could go wrong with that? And now there have been several releases done. I believe, tell me where, I can't remember, were they done in Brazil or somewhere else?
Yes. Brazil and on some small islands in the Caribbean. And I think Tony...
Yeah, Caribbean island. I'm thinking.
Yeah.
Is it Saint something or
Saint something.
yeah? That's a
Saint
you'll
something.
explain a room to play with there.
But we'll see the results hopefully next year.
Yeah.
We'll talk about it. Number 8, TWiV 401, Vector Victorious.
That was yours, Ellen.
Yes.
Was that your
title? Yeah.
Did you
Was
you
my-
picked that one, Kathy?
I
Right?
picked that.
I did. I thought this was really cool. This is a paper from Immunity. Marika Pingen was the first author, and Clive McKinney from Leeds, and they had both been in Glasgow and recently moved to Leeds. But host inflammatory response to mosquito bites enhances the severity of arbovirus infection. So it's been known that something in the bite brings about inflammation, and so it's thought that that's some of the components of saliva. But what they did in this paper was show that there's this recruitment of neutrophils. First, the bite itself causes local edema, so that's tissue fluid buildup, and that retains the virus in the vicinity of the bite, and that then leads to the bite itself leads to chemokines and cytokines being expressed, and those attract neutrophils to come in, and that then and the chemokines allows macrophages to come in, and the macrophage cells actually get infected, and then they go out and disseminate the virus out to the animal. So it's this inadvertent immune response of the edema and the recruitment of the neutrophils that then leads to this recruitment of the macrophages and increases the infection. So it just was really, really a cool story. And she's the one who sent us the pictures of the mouse foot on top of the net, how they did their infections, and then the ear picture with the very precise place where the mosquitoes bit, and you could see the veins getting larger in that ear and so forth. So I just really thought it was a cool story, and it led into some of the next things that I think we're going to talk about, which is animal models. But here, the mice were a really important animal model for doing these experiments.
So number 9 is animal models, and that was also your pick, Kathy.
Yep. So I just highlighted animal models for a couple of things. The Zika virus, the first one that I'm pretty sure that we talked about was Rossi was the first author, and Scott Weaver was the last author on TWiV 383, which was an interferon deficient mouse strain. And then shortly after that, Helen Lazier and Mike Diamond published a paper about a different interferon deficient mouse. But I think we talked about it, but I couldn't find where. But I know Mike Diamond talked about it on the TWiV 414. So those were important to just establish the ability to infect mice. And then in TWiV 392, we had 3 papers that came out all in the same week of pregnant mouse models.
Right.
And so that was interesting because one of them was building on the Lazier Diamond model. And so that was actually from Mike Diamond's lab. Hang on. And then there was one from a Brazilian group that used SJL mouse models, which are a somewhat immunodeficient mouse strain. And then a group that infected the embryos.
Right.
So these first 2, I mean, they're all infecting pregnant mice, but one of them was specifically sticking the virus into the brains of the embryos. I thought that was really impressive. Then I know that I have in my pile of mouse models for Zika a paper about another model, which was in rhesus macaques, but I thought we talked about it, but I couldn't figure out where. But all of these were models that were brought online pretty quickly, although I know the Lazier and Diamond one was in the works before it hit everybody else's radar screen. And then on TWiV 418, we talked about the MERS model. And the reason that a model for the MERS virus is important is because we don't have very many we don't have but one, I think, human autopsy specimen that we can get samples from. And so we understand the pathogenesis very poorly. So having a mouse model is important. And this one was really cool because it's a very engineered model. And the 2 senior co-authors were Mark Heisey and Ralph Barrick. And they made a very engineered MERS model by putting in the DPP, the receptor gene, but just very specific amino acids from the human gene into the mouse. And so the expression of it was not overexpression. And this mouse model, unlike other MERS mouse models, doesn't have the problem of the neurological defects or neurological symptoms that were found with the other mouse models. So that was a really cool model as well. And I recently read a blog post by Vincent about animal models for Zika, and it resonated with me. And so that's why I wanted to make these picks, because, of course, there is no perfect animal model for a human disease except for a human. And we know that we can't do all the experiments that we'd like to do on humans. And so I know Vincent wants to talk a little about this too, but it's so frustrating when people will list as a weakness in a grant proposal that, that this is an animal model and it's not going to recapitulate the human disease. And it just drives me crazy, and I'm pretty much speechless. So you can take over, Vincent.
By the way, the macaque story we did not talk about, but David O'Connor is the PI on that paper, and I'll be going out to Wisconsin and do a TWiV with him
good.
next year. So you can hear about it then. Yeah, I wanted to mention this. So over at Zika Diaries, which is a blog I've been writing for ASM about our work on Zika for the past year, we had submitted a grant application to do some mouse work, which we're very excited about on Zika. And 2 out of the 3 reviewers loved it, and the third reviewer, the main problem was he or she said, "What is the relevance of a mouse model to Zika virus?" And here we've just heard all the advances we've made this year on Zika, understanding Zika pathogenesis using mainly mouse models. And so to criticize, and this was the major criticism that got it above the pay line, as we say.
Right. And
it's just, as Kathy says, it's just ridiculous that you would use this. And I've heard this from many, many other people as well. We all say, they dinged my grant on using a mouse model, which is crazy because they're essential. The other thing, by the way, you can go over to Zika Diaries and read my blog post. The other thing, they said we might not be able to make an infectious DNA of Zika virus, which was very funny to me. Anyway, we've resubmitted it. Hopefully we'll take care of that or get somebody else who doesn't have those silly criticisms.
And this is just a little aside for grant reviewers. When you list strengths, for instance, in the innovation section, if you can't really find any weaknesses, it's okay to put none. Noted.
Yeah.
You could just put none, and...
Kathy, it's funny you should say that because I only had one go around on the study sections when I was active as a researcher, and I heard someone say, and I'm not kidding, it took me 5 different readings to find a weakness in this grant.
Oh,
wow. Can you believe that? And everyone went, they just did that too. It took you 5 times. You read a grant 5 times? My God. It's like if we can read them once.
Yeah.
And how could you possibly be nitpicking that carefully to try to... It was very bad. Very, very bad.
My term for it is gratuitous weaknesses.
That's I think something like that. That's right. That's
when I first came here to Columbia, I wrote a grant proposal to study polio in mice, and I got the same criticism.
Crazy.
What's the relevance of this? It's just crazy. Find something else, because that's not really a criticism.
Exactly.
Anyway, number 10 is virus evolution. I think you picked this, Rich, right?
Mm-hmm. So there were, I thought, a nice group of papers that talked about evolving viruses. Quite different papers, actually. One was the retrospective study of HIV strains from archival samples to look at the spread of HIV globally to see where it actually came from, and the discovery through that the individual who had been cited as patient 0 was in fact not patient 0, and I forget actually what the trace of HIV into the country was, but they tracked it down quite nicely by looking at those samples. We already talked about in TWiV 417, that was TWiV 417. In TWiV 415, we talked about the notion of Ebola evolving, the evolution of the Ebola glycoprotein during the outbreak. We already talked about that.
Right.
And then in TWiV 386, we had on as guests Ted Deal and Welkin Johnson, right?
Yep.
Who had picked out one particular strain of HERV, or ERV, endogenous retrovirus, and traced it back through many eons and looked at its evolution, which was an in silico project using databases. And I thought it was just fascinating what they did. And they traced it. They saw it in quite a number of different species and doing cross-species transmission and et cetera. And it was a really nice study of the evolution of one virus.
Yep.
So those were good.
All right, I think I went through the titles to find the best one. I think the best one is Zika and the Guys with Diamond.
That's good.
And a second, the runner-up is Boston Quamins.
Yeah.
I like it a lot. And my favorite meme of the year, which actually was sent in by a listener, one does not simply walk into the placenta.
Yes.
Finally, my prediction for 2017, there will be virus infections. In fact,
really?
in the oceans, there will be 1 quadrillion infections per second. That's 1 followed by 24 zeros.
And they will infect them in the hills and on the beaches and in the mountains.
That's right.
What's that from?
That's Churchill. I'm misquoting
that's
somewhat.
Right.
But,
that's right.
We will never surrender.
All right, this is our last Curiosity Stream supported episode for some time, I guess. Curiosity Stream is the world's first ad-free nonfiction streaming service. They have over 1,500 titles and 600 hours of content, founded by John Hendricks. Used to be with Discovery Channel, and that means you will get real science. You can watch it in your web browser. You can watch it on your TV using a Roku or a Apple TV or similar devices. 196 countries worldwide. What they have is a wide variety of science and technology, nature, history, interviews, lectures, things like Stephen Hawking's favorite places, where he pilots a CGI spaceship across the universe, stopping at his favorite places. I like that one. I watched that recently. There's a 3-part series by Derek Mueller, creator of Veritasium, where he explores online safety and security. Deep Time History, a 3-part series on the history of the universe, and Underwater Wonders of the National Park. So just a couple of the cool episodes that they have. They also have a lot of super high-definition content, so-called 4K libraries, over 50 hours of that. They do have monthly and annual plans available, starting at just $2.99 a month, less than a cup of coffee, or a single title on other platforms. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That is 2 months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support this year of TWiV. Let's do a couple of emails here. Let's have Kathy take that first one, since she likes this topic, I think.
Patricia Wright. Hi, TWiVolios. I actually don't have a science comment for you this time, just 2 follow-ups from the last 2 team episodes, 417 and 418. Number 1, thee versus thou. I guess science fiction junkies aren't always also fantasy alternate history junkies, although I suppose if one spent a lot of time reading a Bible, one might also have internalized how these words work. Thou is the familiar form of you, singular, used as the subject of a sentence. Thou art my best friend, or couldst thou pass the peas? While thee is the familiar you, used as the direct/indirect object of the sentence. Hark, I will pass the beans to thee. Thy and thine, incidentally, are roughly equivalent to your, as in peas, and yours, the peas are yours. Modern English has merged thee and thou into the formal you for all instances, which is why we've always said your majesty, not thy majesty, but has kept the same formatting for things like she versus her. For example, to tie it all into a common TWiV theme, though, we really suffer for a lack of the plural you like German has. I'm for y'all and all y'all. I started using them when I learned German and realized how useful it was to have a way to distinguish between one you and many yous. Anyway, number 2, you asked what podcasts we listeners also listen to. I've sent in recommendations for picks before, but here are some more. The History of Rome and its spiritual successor, The History of Byzantium, as well as the Revolutions podcast done by the same podcaster who did History of Rome. I'm getting a kick out of The History of English, might be explained by the same love of fantasy and language that makes thee and thou make sense to me. And also the British History podcast. I enjoyed both iterations of Serial that Rich mentioned. I listen to NPR's TED Radio Hour, and I periodically peruse BBC for educational podcasts like their Life Scientific that I mentioned before. And of course, I'm addicted to the Twixcasts and Audiomunity. Better get Kathy to say that one. That's all for now. Happy sciencing, everyone. Patricia. P.S. In one of your Curiosity Stream ads, yup, I listened to them, you refer to Edward Snowden as a former director of the NSA, probably a slip of the tongue. But just in case, I figured that should get cleared up. He used to work for the CIA, but I've not ever heard of him directing any particular government agency. Patricia.
Yeah, Snowden was an NSA contractor which is
Yes.
right in the Curiosity.
Not director, contractor.
It's right in the Curiosity Stream copy. I misspoke. Yeah. What's the German plural for you? Does anyone know? Is it ear? IHR?
I think I'm wondering about this. I should know it, but I think ear is correct.
Okay.
I was
We'll
hoping it
get
would
mail
be y'all spelled with a J.
Well, I think it depends on what part of speech the U is.
That's a cheese. I thought it was Jarlsberg.
Oh.
I know.
No,
Just
that's
end
your
of
town.
year, get it out of my system, that sort of thing.
Dickson, can you take the next one?
Sure.
So because
Sorry.
So Z is one of the U's, Z capital SIE.
Correct. That's
And-
right.
And so ear is
And that can be plural.
right. It's complicated. There's lots of these forms.
It's complicated.
Yeah.
And Scalco writes, I enjoyed the discussion about proper terminology for cells infected with silent Ma virus. Though you might be interested in a term coined by Jan Svoboda in 1963. He isolated rat cells transformed with a strain of RSV, or Rouse sarcoma virus. No virus was produced by the rat cells when in culture, but virus was produced if the cells were injected into chickens. He called these rat cells virogenic, with lambda lysogenic in mind, but mindful of the distinctions. By the way, his name Svoboda is, I believe, Hungarian, and it means freedom. How would I know that?
Oh, boy.
Because the center fielder for the 1986 Mets was Ron Svoboda,
Yeah.
and people kept referring to what his name meant and all that.
Yeah,
thing.
it does mean freedom.
So there you go.
Check.
He was a Czech. That's true. That's not Hungarian. Czech. Sorry about that.
Oh, you're bound to get something wrong.
I try hard, actually.
Go ahead. Finish the email.
I'm learning a lot about my retrovirus book.
No. Read that again, please.
I am learning a lot. Writing my retrovirus book, and she puts a little happy emoji there.
So Ann Scalco, of course, was one of my co-authors on Principles of Virology.
Nice.
She's retired, and she's writing. Oh, maybe I shouldn't say that, Ann. Okay, you're not retired, but you're writing a retrovirus book.
Yeah, right.
And then just someone.
I was glad to hear that she was interested in what was a thoroughly pedantic discussion of the terminology.
No, I called it a lysogen, and you were right to say, but that's a phage terminology. And here we have someone who thought about that. I think that's cool, virogenic. But Ann is someone I didn't think listened to TWiV, so I was surprised to see that.
There you go.
Let's see. I will take Mohammed's next. Dear TWiV team, this is Mohammad, your MERS coronavirus correspondent from Saudi Arabia. I would like to congratulate you all for a very successful year. Not bad for an even year, Dr. Racaniello. Wish you all a more successful 2017. 2016 carried great TWiVs as usual. Personally, the interview with D.A. Henderson was the most inspirational and ranks highest in this season. As requested in a previous episode, other podcasts that I listen to frequently include Nature Biotechnology Podcast, Hidden Brain, Radiolab, and most podcasts produced by Microbe TV. I have two follow-ups. One on TWiV 420 entitled Orthogonal Vectors. The use of the term orthogonal was discussed. I would like to add one more example for its use applied through regulatory settings. Regulatory agencies use this term frequently in the context of describing the need of use of a second, third technique to confirm the findings of the first one. For example, protein aggregation, a nasty protein instability problem, is typically evaluated by manufacturers using multiple technologies, size exclusion chromatography, dynamic light scattering, and analytical ultracentrifugation as an example, where two techniques are used orthogonal to the first one. And it gives two examples in links. My second follow-up concerns episode TWiV 413 entitled Partnerships, Not Parachutes. In this episode, the many advantages of using camel vaccine against MERS coronavirus were discussed. However, this discussion was followed by skepticism by some of your esteemed guests about the acceptance of such vaccine among Saudi people, especially breeders of racing camels. Personally, I don't see how they came up with that conclusion. Let's see what the WHO statistics say about Saudi citizens' compliance with vaccines. He provides a link for that. DTP, diphtheria, tetanus, pertussis 3 immunization among 1-year-olds is nearly 100%. Measles immunization among 1-year-olds, 98%, compared to 96% in the Netherlands and 91% in the U.S. 2 points. 1, Saudis strongly believe in the power of vaccines. The scars caused by smallpox are still seen in the faces of the elderly. 2, I really don't think Saudis love their camels more than their newborn babies. About the racing camels issue, let us suppose for the sake of argument that no racing camel owner agreed on vaccinating his expensive camel. Those kinds of camels are rare. They are expensive for a reason, and I think herd immunity would take care of that. This brings me to the recently announced news about a MERS corona vaccine on trial in camels going to be held soon. It provides links. The vaccine was developed in the Jenner Institute, the University of Oxford, through a joint collaboration with King Abdullah International Medical Research Center, Riyadh, Saudi Arabia. The dates of the trial were not announced yet, but I guess it will begin within the next 2-3 months. I will keep you posted with the results. That's interesting. I actually sent your comments to the panelists on that TWiV episode at Boston University. I didn't hear anything from them, though. It was a while ago, too. Now, let's switch gear to a more serious matter. Weather. It is snowing in Saudi again for the third consecutive year. See attached pictures. Not only that, but we officially have lakes now. See the following YouTube for a newly formed one. And he sent a bunch of pictures of snow and a camel with snow on it. And the funny thing is, go ahead.
I was just going to say they're very artsy pictures of the snow.
They are.
Wow.
Yeah, they're very nice.
Yeah, even NASA's website had a few of them.
The cool thing is, in our last yearly summary a year ago for 2015, Mohammad also wrote us and sent pictures of snow there, and I used one of them for the episode image, so we'll do the same. One last thing.
The camel one.
Camel is really cool. We'll use that one.
Now, is the snowman figurative art?
Which one is that?
There's a snowman in front of some mountains in these.
I don't know.
Okay, just curious.
I don't know, Mohammad, but the one with the highway and the mountain.
It's quite remarkable.
Amazing. It was amazing.
Right. No, I was just wondering because there's a Muslim prohibition on drawing figures, and I don't know how devout somebody would be to make a snowman there, or if that's permitted.
Yeah, I don't know, but the one with
it probably doesn't come up very often in Saudi Arabia. But the one with the camel with the snow on it and the mountain, the purple mountain, those are really cool.
Yeah, this one with snow on the dunes, which is amazing also.
Yeah.
One last thing. Many moons back, the TWiV team used to leave us 30, 60-second segment at the very end of most episodes where we, the listeners, could hear the team discussing the title of the episode. I really miss that and wish this customary act would come back again, at least for the very creative ones. Just a thought. Anyhow, extremely sorry for the lengthy email. I just had so many things to talk about. My name is Mohammad, and I am a TWiVaholic. That's great. I love it.
Yes.
I just love it. Yeah, I stopped because the episodes were getting longer, and
yes,
I didn't want to add more.
But that would reward those people who listen to the end.
Stick around.
Yeah, we'll do that. And maybe they just weren't all that creative, because most people nowadays are saying, yeah, I like that one. Yeah, I like that one. Yeah, I like that one. Then someone has to decide. But when
anyway, Mohammad, we call them Easter eggs. That's the term for them.
Little snippets at the end of the show that you wouldn't find unless you listened all the way through.
Let's do just two more. Alan, can you take the next one?
All right. Len writes, Hi, Vince. I listened to TWiV 419, and during your discussion of the current biology article about the mummy that had smallpox DNA, I remembered a book from a few years ago called Pax Americana, The Great Smallpox Epidemic of 1775 to 82 by Elizabeth Fenn. Apparently contemporaneous with the American Revolution, there was a massive epidemic of smallpox that covered most of the Western Hemisphere. Anyway, I thought you guys and your listeners would find it interesting. Cheers.
Right.
Yeah, it's absolutely right.
You know, when they did a documentary drama on John Adams.
Yes, I saw that. Yep.
The kids, some of his kids caught it.
And that's when they were using scarification to try and prevent it, with an understanding that, it could kill you. You got about a 30% chance of dying.
Yep.
Also, this would, Rich, do you remember we did that brief paper on the sequence of smallpox from that mummy, right?
Yeah, that was. I'm still struggling with that. Right,
you working on it?
It just because it really overturns a lot of thinking about the emergence of smallpox, and I'll be interested to see how that pans out in the long run.
But I think this outbreak during the American Revolution would fit in with the. That particular strain that was found in the mummy.
Fine.
Right? Yeah,
that works fine.
That's no problem.
Matter of fact, you can go all the way back. I think my dating is that, one of the most dramatic outbreaks was Cortez invading Mexico.
Yeah.
Okay,
Mexico,
and bringing smallpox and the Aztecs getting wiped out. I don't think there's any question, but that was smallpox, and that was 1500, and I believe that fits into this as well.
All right, last one, Rich.
Chris writes, "Hello again, TWiV team. I fear I may be a bit late to be lucky number 23, but I know that I cannot win if I do not play." The weather here in central Ohio was a very nice spring day, with the high hitting somewhere just short of 20 degrees C with an hour or so of full sunlight. It recently finished raining, and the temperatures are supposed to fall back to a warm winter for the rest of the year. Mm-hmm. Anyway, I was inspired by the podcast recommendations in episode 421 to share some of my other favorites outside of the Microbe TV family. If you only have time to share one, Far Lands or Bust is the one to pick. People Behind the Science, interviews about the lives of current scientists. I mentioned this in a previous email. If my sent email folder is accurate, that sounds interesting.
Mm-hmm. I think we had that as
Far Lands or
yeah, I believe so. Yeah.
Okay. Far Lands or Bust, a YouTube series of a man's journey to the end of the world in an old version Minecraft. It now comes in both podcast and video flavors. Host Kurt J. Mack discusses gaming news, answers to listener questions, and often the best part, discuss space news. Kurt is an avid fan of astronomy and rocket launches, so both the launch itself and the details about the mission are discovered whenever they happen. This series is a long-term fundraiser for Child's Play Charity, which gives toys, books, and games to children in hospitals. I gave this the longest description because after I somehow found Flob around 5 years ago, I followed some of the people that Kurt recommended, and that started a long chain of recommendations that eventually led me to the TWiV and podcasts. Track changes. Two guys discuss the life of a company they founded in New York City. It has little relevance to my life, but the excellent chemistry between the hosts and short episodes keep me listening to each new episode, and the topics often turn out to be surprisingly interesting. Accidental Tech Podcast. Three guys discuss the tech industry, but mostly all things Apple. The Scientific Odyssey. It's all about how we know what we know as science came into existence. Currently, they are discussing the shift from a geocentric to a heliocentric model of the universe, and more broadly, how science began to emerge as a distinct discipline from natural philosophy. That sounds interesting as well. 99% Invisible. They tell interesting stories about all kinds of things that you would likely not know about, so it's a surprise every week. Even listening at 2x speed, sometimes I get more new episodes in a week than I can keep up to date on, so I have to pick and choose which episodes to mark as played and which to actually listen to. My usual triage is short, less than 1 hour episodes, Twix episodes, usually TWiP followed by others. 3, which ones have the most interesting show notes? Thanks for giving me all these great podcasts. Chris, very nice.
Got some good ones in there. Yeah.
Yeah, boy.
I do listen to Accidental Tech Podcast myself. I have to tell you, this morning they were discussing one of the podcasters, John, just bought a new car with a manual transmission. And when he finished talking about it, one of the other guys said, "You and your gears." And so John said, "Well, your car has gears too. You just can't change the ratio." And they went through this long argument. And one guy said, "No, I don't have gears. I just have one." He has a Tesla. He says, "I have forward and reverse." And John said, "No, you have gears. You have those little circle sprockety things underneath. Definitely you have gears." And then finally the guy said, "You're being pedantic." And I said, "Oh my God, another podcast being pedantic." It was really fun.
Well, now electric motors have a very different power curve from gasoline or diesel motors, and I'm wondering if Teslas actually do have more than one set of gears.
There
may be like a reduction gear down to the drive shaft, but other than that, it may be possible to run the entire thing in the same gear.
Sure.
Yeah, I'm sure that happens, but there is at least one gear.
That's why they have such remarkable acceleration.
And
there's always going to be at least one.
So gear. John is a very, very, he's a pedantic guy. And, so you have a gear. It's only one, and you can't change.
So, yeah, as a matter of fact, if you Google, there's a Tesla forum here. It says there is a single fixed reduction gear to take the motor RPMs down to axle RPMs. And beyond that, electric motors are very efficient, so they heat up very little for the amount of power they're putting out. So I guess there's no real transmission.
And by the way, wait till you see what he comes up with next.
Yeah, what's he going to
He's working out on a car that's going to be quite remarkable.
What's it going to do?
It's called a V something, as I recall. It's got incredible power and acceleration. Yeah. Way beyond the current model.
Okay.
That's not going to help you when you're stuck in traffic.
Tell me about it.
Let's do some picks of the week.
Wait.
Yes.
The suspense. What? Is Chris number 23?
Oh, no, we didn't reach it yet. I'm sorry. Yes.
Okay.
We're not there. I've had some responses, not that many, because I think it's holiday week and a lot of people are not listening, but we're not there yet, so keep on trying. And we will have more giveaways in the next year. I have a brand new bunch of different kinds of virology books that I get from ASM, and I'm going to give them away. Same thing on TWiM. So listen to both. And of course, over on TWiP, you can get Dickson's book for free. You just download it from parasiteswithoutborders.org, right, Dickson?
You got it.
Let's do some picks. Alan, what do you have?
I have a fun article on Ars Technica, and I see you picked something from Ars Technica as well. So they're definitely getting the TWiV bump today. This is about the city of Cahokia, which you haven't heard of because it's underneath what is now East St. Louis. There were tribes of Native Americans and civilizations, actually, that built up in the Midwest, extending all the way down into the South. And we don't know a whole lot about them, except that they tended to build these large mounds of earth. And you can go in the St. Louis area, and they're more down all along the Mississippi River Valley. You can see these structures that are there. And this story is talking about the excavation of a major city that was on this particular spot that had these mound-like structures and big open courtyards. And they estimate that the city in the year 1050 AD had a population of 30,000 people.
Really?
Which meant that at that time it was bigger than London or Paris.
Wow.
Interesting.
And there's no written record of this time, and there are some oral traditions from Native American tribes that are, in or have been in that area more recently, but they don't consider these folks to be their ancestors. So there's a lot of mystery about why this civilization came into being and what the basis of it was. It was clearly agricultural. But then the other big mystery is, why did it go away?
Mm-hmm. Yep.
And the story is really well done and explaining the complexity of excavating this thing and also the scale of what it was like. My own personal theory is that there was some kind of epidemic, but who knows?
Yeah.
Well, they're in a perfect position to be washed away, too, by some major floods.
Yes.
Maybe not the buildings, but the people.
Beginning in the late 900s.
Right.
No, city's apex in 1050. I'll be darned.
It's cool. Yeah. They have some skeletons here, and they have teeth. They could try and get some pathogen DNA out of them.
The skeletons are from apparently some sort of ritual sacrifice.
Wow.
Yes. And as the population declined, apparently it went through this series of transitions, and the article talks about how the city plan changed and what that might have meant. But just a really, really cool story.
Wow. That's pretty cool. I like it.
Yeah.
Rich Condit.
I have to tell you how this pick evolved. Briefly.
Are you sure it evolved? It's kind of a passive thing, you know.
At any rate, yeah. So you can tell me when we're done here. So believe it or not, last night my daughter was showing me how she was cleaning the grout in her kitchen with a toothbrush, and she was using as a cleaner a mixture of baking soda and vinegar, which she had heard was, a very good thing to use. Okay. And as a matter of fact, if you go on the web, you see it all over the place. Oh yeah. Best cleaner from household items is vinegar and a mixture of vinegar and baking soda. But we started to question this. Say, I wonder. Okay. So we looked into it a little further, Googled it around, and I wound up on this site that is written by Sabrina Stierwalt, who is an astrophysicist at the University of Virginia, with a passion for communicating science, it turns out. And she's got a bunch of small podcasts. And she runs a podcast with small episodes called Everyday Einstein. And she had a rap about this vinegar and baking soda thing. She said, apparently people decided that since each of them were good cleansers by themselves, a combination ought to be dynamite. Mm-hmm. But in fact, it doesn't make any sense because one of them is sodium carbonate and the other is acetic acid. And you mix the two, you get water and sodium acid.
Yeah, exactly right.
which
neutralize each other.
Worth anything. Yeah, right. So it was very nicely written, very accessible. So I looked through some of her other stuff, and I just kind of wanted to give her a bump because she really does have a passion for communicating science in just a tremendous variety of areas. So she's got a little she's got a little TEDx talk as well, where she talks about what science is. And she talks about the role of curiosity and serendipity in science and how we use it all the time. So she's a real advocate, and I wanted to give her a shout out.
Oh, nice.
She's got a little podcast here. Everyday Einstein. It's about 10 minutes or less each episode.
Yeah.
It's got one on CRISPR, too.
Yeah.
Okay, it's cool.
So I should follow up by saying, so we did the experiment.
And?
We did a streak of grout with vinegar and a streak with baking soda. And my 5-year-old grandson suggested we do the control right next door with the two together.
Your 5-year-old grandson suggested that?
And I suggested we also do one with just water.
Mm-hmm. Right.
Absolutely.
And our conclusion is that the most important ingredient, to quote my daughter, is elbow grease.
Yeah, that's the toothbrush probably. Let's do it without toothbrush.
Dickson, what do you have?
Yeah, well, I've got a retrofitted Hubble Space Telescope that got a brand new high-definition wide field planetary camera. And they re-examined the Eagle Nebula for pillars of creation. And this is a video of an astrophysicist explaining what all of this means in terms of star formation. You can now actually see the nucleus of a star inside these clouds, which you couldn't do before. And he's got them side by side so you can see the big difference. So the more you see, the more you know. I put this up on purpose. I put this up on purpose because of the rhetoric that's going on right now with regards to the incoming administration to poke out the eyes of NASA by discontinuing funding for Earthwatch, which absolutely has me apoplectic. I can't respond to this intellectually at this point. If you can't see something that's going on, then you can't complain about it, like, pollution and stuff like that. So this is a great example of what happens when you can see more. And I picked it for you too, Rich. I picked it because I knew Rich would at least like it.
I watched not all, but most of this video, and it's quite good.
It is.
Very good.
I also watched most of it. And then we got into new microphone mechanics.
Right. I'm sorry to say, Dickson, that I don't think any member of the new administration or the old one listens to TWiV.
Well, the old one will have a lot of time to listen to.
I would put a larger bet on someone from the old administration listening to TWiV than
perhaps
the new administration. But yeah,
Kathy, what do you have?
I picked something that was suggested to me by Mike Imperiali. It's called the Riley Top 10. And Mike wrote, at the end of every year, the Riley Center for Science, Technology, and Values at Notre Dame posts its top 10 list of ethical dilemmas and policy issues in science and technology. They're not all biology, but I think it's pretty cool and provocative. And so when you go to that site, they have things about a car that may be able to read your emotions and adapt to you. Something called the self-healing body. So things that you would swallow and so forth. What was the other one? Brain hacking, how you can do your own EEGs. And so what that might lead to when all that data becomes available. It's been a couple days since I looked at this. So anyway, it does have it has a short blog post about each one and describes what the issues are, and then at the end has a kind of nice set of provocative questions about it. So I didn't know about this policy center at Notre Dame, and evidently they do this pick at the end of the year. So I thought at the end of the year, it's nice to have another end of the year pick.
Cool.
So. All right. My pick is an Ars Technica article called, which was pointed out to me by Dickson. So thank you, Dickson Despommier.
You're so welcome.
Naples' sleeping volcano might be waking up. So there is a caldera, a collapsed crater near Naples. It's called Campi Flegrei, that formed 39,000 years ago during an eruption, and it seems to be having some activity. So I wrote in the notes Vesuvius, but it's not really Vesuvius. It's a different thing. But there's lots of volcanic activity around.
Italy's famous for it.
Naples, there's some in Sicily as well. But they say you can't really tell. There's some signs that things are happening in terms of gas release and so forth. But it won't be. They say it won't be an eruption as violent as the one that it began with. It's erupted over the years a few times as well. So this is near Naples. It would be interesting if something happens, but we don't know when it will happen. But I think it's an interesting article and gets you thinking about volcanoes, which you don't talk about much here on TWiV.
Interesting, meaning frankly terrifying for the people who live near this thing.
Frankly terrifying.
Well, it may not be that bad.
It may not be bad, right?
There is actually, this is based on an academic article in Nature Communications, which has a title that is less engaging. Magmas near the critical degassing pressure drive volcanic unrest toward a critical state.
Yeah, I guess most of the people of Naples didn't catch that one in the local newspaper.
Probably not.
Wow.
I get a kick out of these titles, by the way. It's just another aside. I get science delivered at home, and my wife is not a scientist, and every now and then I'll say, honey, did you know? And then I'll read the title of some, a material science article about the rotatory spin of a chiral molecule that has and she'll say, oh my God, how did we miss that?
So this their abstract is actually very useful. During the reawakening of a volcano, magmas migrating through the shallow crust have to pass through hydrothermal fluids and rocks. The resulting magma-hydrothermal interactions are poorly understood. Here we use the results of physical and volatile saturation models to demonstrate that magmatic volatiles released by decompressing magma at a critical degassing pressure, CDP, can drive volcanic unrest. We show that here. We propose that magma could be approaching the CDP at Campi Flegrei, a volcano in the metropolitan area of Naples, one of the most densely inhabited areas of the world.
Indeed.
Which would magnify its impact.
Would magnify its impact.
That's right.
Indeed.
Boo.
So we had a listener pick from John, which was an episode of Radiolab, which I think Rich already read before.
Right, I just copied that in.
Times they are a-changing. All right, that is TWiV 422. That's it for 2016. It's over. Almost over. A couple more days. You'll be able to find us at iTunes and at microbe.tv/TWiV. We love getting your questions and comments. Please send them to TWiV at microbe.tv and consider supporting us. Go over to microbe.tv/contribute. We have several ways you can do that, including a Patreon account. And why do we want you to support us? Well, one of the things we can do is travel together and do shows together, and like we did for number 400. And we'll try and do more of that next year. And, of course, my goal is to try and get some technical assistance here, because I would like to start a couple more podcasts next year, but I do need some help to do that. So check that out.
We also get some pretty cool equipment.
That's right. Kathy just got a new mic and a new headset, and she sounds better.
And I have a holder for my iPad.
Yeah, Dickson has a holder for his iPad.
Now I can read.
Doesn't have to stack it up. And we'll get Rich a new microphone. Alan got one earlier this year. Yep. And we will always try to improve the TWiV experience here on TWiV. Dickson,
I'm going to miss you guys.
Where are you going?
Well, you're not going to be here until all of next year. I mean, come on.
Yeah, I know. It's a long time. Dickson Despommier is here. He's an emeritus professor at Columbia University. He's also at parasiteswithoutborders.org and thelivingriver.org, among other websites. Thank you, Dickson.
You're welcome, Vincent.
Always good to have you, despite your errors in management.
It's always good to be here, no matter what.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun, and I want to wish everyone a Happy New Year.
Yeah, and from us, too.
Rich Condit is a emeritus professor, formerly at the University of Florida in Gainesville, now in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Great fun.
And Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this show, Curiosity Stream. Happy New Year, everyone. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.